Welcome back to the course Fundamentals of General Immunology. This is lecture number 5. The title is Self, Non-Self and Danger Recognition Models. So the term self stands for self-antigens, and these antigens are any molecules that are produced by the body's own cells that act as an identification marker. That means these are the proteins which are produced normally by the cells, and they should not react the body should not react with those antigens or those protein as a foreign. So these are considered as self. Coming to the function, they are recognized by the immune system as a self. When it comes to self, the immune response does not produce against those antigens, which can lead to some harmful reactions in the body. And the basic examples of those self-antigens are major histocompatibility complex, which are in short called MHC molecules. Also the blood group antigens. These blood group antigens are either A, B, O, and also the proteins which are normally expressed on the surface of liver, heart, brain cells, also in kidney and other tissues of the human body. So these basically the proteins which are normally produced by the body system, and they are not considered by the body immune system as a foreign. What could happen if there is a failure to recognize this as a self? Sometime it may happen that the body recognize these self antigen as foreign, and in that case they raise an immune response. And if there is an immune response against the self antigen, then the condition is generally considered as autoimmune diseases, which is the immune response against the self antigens. When we call this that why there is no production of immune response against a self antigen, the terms comes to the context of these biology is called tolerance. So tolerance is a very specific term where antigens are not recognized by the body as a foreign. Quoting an example here, that we take every day the food particles. Now food particles are coming from outside. For example, the rice, the other kind of breads and cereals, these are coming from outside. So considering the point that they are coming from outside, they should not consider as self, and therefore these are important thing for the human body, and therefore body should not react against those materials coming from outside. So there is a term called oral tolerance, where the body can recognize these molecules which are entering into the body cavity through the oral, and they are not raised, the body is not raising an immune response. So essentially those are considered as tolerance. Since they are considered in the level of oral, then they are called as oral tolerance. So these are some of the examples of and some of the concepts related to self antigen. Now onwards in this chapter, whatever we will read apart from self is actually non-self. So any protein which are coming from outside in any form is considered as non-self. So when we say that antigens, actually, so antigens is specifically the molecules which are foreign in nature. In immunology, generally antigens are foreign in nature. They can trigger an immune response, sometime very large immune response, in order to remove that external stimuli. The examples of these antigens could be exogenous antigens. The examples are bacteria, pollen grains, toxins secreted by some bacteria, toxin coming from environment entering into the body cavity. These are all considered as exogenous in nature. So for example, suppose if this is a cell and this is the nucleus of that cell, and this is cytoplasm. So suppose this antigens which are coming from outside, this could be in the form of a bacteria, maybe gram-positive or gram-negative bacteria, and this can enter inside the body from outside. So therefore these are called as exogenous antigen, since they are coming from outside. This could be a toxin and pollen grain which can coming from outside. While once they come from outside, they can always go and bind to the cellular receptor which are present on the host cell and then internalize through either receptor mediated endocytosis, or they can passively enter inside the host cell also sometimes. While in case of endogenous antigen, endogenous as name itself, the antigens are produced inside the cellular system. So in that case, the classical examples are viruses, the infection of the virus into a cell, susceptible cell population. So suppose this is a viral molecule which is targeting this particular cell type. So this virus can bind to the receptor present on the host cell, and after binding they enter inside the host cell by receptor mediated endocytosis. So suppose this virus is having a genome, this could be a DNA or RNA but never both. So this virus post infection coming into the cytoplasm, and this nucleic acid which is present inside the virus is released into the cytoplasm by various mechanisms. We will not go in detail about those, but ideally to understand this endogenous antigen, we need to understand that this nucleic acid coming from outside can form RNA, messenger RNA, and these RNA can form the protein. So if we see this phenomena, the virus is coming from outside, but then after coming from the outside environment, this is entering inside the host body and particularly in the cell, producing an RNA which is in turn forming a protein. So essentially this protein is formed inside the host cell, and therefore these are considered as endogenous antigens. As I said, all the viruses, most of the viruses are actually producing endogenous antigen because viruses can't produce outside, can't reproduce outside. They are facultative. Viruses are obligate intracellular parasite, so they can always infect the cell and produce the protein inside the cell. There are examples of bacteria which are considered as endogenous antigen. For example, some of the bacteria are example is for example, the bacteria such as Mycobacteria tuberculosis is actually an example of the pathogen which is intracellular in nature. So the Mycobacteria tuberculosis is an example of intracellular pathogen. So the protein which are produced post infection of Mycobacteria tuberculosis is always endogenous in nature. So these are two classical examples of exogenous and endogenous antigen. Then comes the autoantigens. Auto stands for self. So these are actually the antigens which are produced by the host cell itself, and sometimes they can be produced in a way that the body immune system can consider these as a foreign. For example, lupus and rheumatoid arthritis are the examples where the body immune system can target its own antigen as a non-self, and therefore this can lead to the condition such as arthritis in the human body. So these are examples of autoantigen which are body own proteins which can consider as foreign in nature. So these are the three examples of exogenous, endogenous and auto antigens. So next is the antigens which are expressed as a marker in the tumor cell. Now the cancer cells are unique in terms of many aspects as compared to the normal cell. So if the cell is actually cancerous in nature, this cell will express lot of protein on their surface. These are considered as marker protein. These are not normally expressed in the normal cell population, and therefore this can be considered as unique for the cancerous cell, and this could be a marker also. Each cancer cell type will express a separate protein which can be identified, used as an identification marker for that particular type of cancer. For example, if the cells of pancreas can express a unique kind of a protein on cell post conversion of its cancerous form, we can call that a protein is a marker for pancreatic cancer, and similarly for other cell type as well. Now if we see the classification of tumor related antigen, there are two classifications. One is called tumor specific antigen or TSA, and the other is called tumor associated antigen. So tumor specific antigens are unique proteins which are present in the cancer cell, and these are formed by the mutation of the host, mutation generated in the host cells. For example, there is a protein called P53, which is actually a tumor suppressor protein. The protein can inhibit the tumorous growth in the human body, and also the RAS, which is a rat sarcoma protein. These are actually a host protein. Sometimes it may get mutated. Mutated stands for the protein may change its functionality because of the change in amino acid structures or the change in the gene pattern, and therefore this could lead to different kind of cancer. Many cancerous cells have been reported to show or exhibit the mutation of P53 or RAS proteins. The second example is BCR-ABL protein fusion. So this is a very unique fusion that could lead to blood cancer. So BCR-ABL2 is another example, and this is a very unique fusion protein that can lead to chronic myeloid leukemia. The full form of BCR is breakpoint cluster region, which is given in the bottom, and also ABL1 is Abelson leukemia viral oncogene homolog 1, it's in short called ABL1. Now, these BCR and ABL are expressed in two separate chromosomes. So I could give a diagrammatic representation here. For example, there is a chromosome short arm and long arm, and this is the location of the gene here called ABL. This is in the chromosome number 9, and then we have BCR, which is located in another chromosome, chromosome number 22. So this is another protein called BCR. Particularly, the shuffling of the movement of this fragment into chromosome number 22 is reported to be very toxic. So the relocation of 9 to 22 can lead to chronic myeloid leukemia, which is a blood cancer, and this type of translocation is called Philadelphia syndrome. Philadelphia syndrome. So this is just a translocation of 9 to 22, from 9 to 22 chromosome number. So apart from these, there are specific proteins which are produced by viruses. Now, emphasizing the point here, there are many viruses that can cause cancer. For example, human papilloma virus, HPV. There are other viruses which can also cause cancer, such as HIV. The viruses which can cause cancer can express many different kind of proteins which are expressed on the surface. So for example, the human papilloma virus can express E6 and E7 proteins which can lead to cervical cancer, and the cells which can be visualized for detecting cervical cancer should express E6 and E7, which is an indication that the person might be suffering from HPV infection that can lead to cancerous progression. So these are some of the examples of tumor specific antigens. So these are very specific for tumor. Coming to the next one, which is called tumor-associated antigen. So these are actually a normal protein. So remember, these are actually the host protein which are either overexpressed or abnormally expressed. That means there are optimal concentration of these protein which could be present most of the time in the cellular system. But if there is an overexpression or because of the abnormal expression stands for the odd expression. Sometimes the body may not need the expression of those protein. If there is an expression of those protein in some odd time of a life cycle, then we can consider this as an abnormal expression. Give an example that in the early phase of life in a human being, there are proteins which are required for the growth of a cell. Now these proteins are expressed in a very large quantity in a very early phase of a life. Later on, say for example at the age of 35 or 40, human may not need that protein, and for that reason those proteins are not expressed at that time. If by chance those proteins start producing at the age of 35 or 40 for the growth promotion, then those proteins can be considered as abnormally expressed, and this could lead to cancerous progression. So this is what we call tumor-associated antigens. The classical example is the human epidermal growth factor receptor 2, which is called HER2 receptor. These are overexpressed in case of breast tissues and also in ovarian cancer. Prostate specific antigen, these are also a marker for prostate cancer. Then what I discussed is actually carcinoembryonic antigen. Embryonic stands for the one which is developed early phase of the embryonic development, and once they are not in the produced, once these embryonic stages are over, then these antigens are not supposed to be produced, and if they are produced in the later time point of a human life, then they can lead to different forms of cancer, and that is what you call carcinoembryonic antigens. And the last one is called MUC1, which is standing for mucin. Mucin is also a very specific proteins which are secreted by the mucosal layer, and these can also overexpressed could lead to a particular kind of carcinoma. So the carcinoma condition may show an overexpression of mucin like protein. So these are some of the proteins which are related to tumor, either tumor specific antigen TSA or tumor associated antigen TAA. All right. We'll move to some more definition related to antigen. One such name that comes very often in antigen biology is called haptens. Now haptens are actually a very small molecule, and this small molecule may not trigger an immune response by its own, but when they are added with something else, for example, this is a haptane molecule, these alone may not produce an immune response, but when it is added with some other carrier protein, for example, if this is the carrier protein that got added into this haptane, and this protein, so this combination of this protein with this haptane may raise an immune response. So that means this may not be alone immunogenic in nature, but once it is coming in contact with a carrier protein, that may lead to immunity. Apart from that, the RBC, which is what we call red blood cell, is expressing lot of sugar moieties on its surface, and these are the very strong glycoproteins which are expressed on the surface of the RBC, and this sugar marker can define the blood group. And the examples are A blood group, B blood group, and also O blood group. These are nothing but the expression of the sugar, specific types of sugar on the red blood cell that can define the types of blood group antigens. Apart from that, we classify antigen based on its reactivity against the T cell population. So as we know that there are two types of lymphocyte, one is called T and the other is called B, and T cell specific immunity is critically important for generating a cell mediated immunity or cell mediated immune response. So sometimes there are antigens which are dependent on T cell processes. Once the T cell will process, then those antigens can induce an immune response. So these are actually dependent upon T cell. So therefore these are called T cell dependent antigen. And then there could be a cell which may not require T cell activation. They can directly activate B cell to produce an immune response, and therefore these are called T cell independent antigen. So this could be a dependent and this could be independent antigen. So there are ways to define the antigens, and just one thing which I wanted to emphasize here, that antigens are actually the term given to proteins which can generate an antibody. So we can call it antibody generator. We have emphasized that point in the earlier classes as well, that antigens are very important proteins which could lead to an immune regulation, but not all antigen can produce an immune response, and therefore the term in recent years changed to a term called immunogen. So immunogen is actually a term that is related to any antigen that can raise an immune response. Otherwise those antigen may not called as an immunogen. So there is a difference between antigen and immunogen. So immunogens are any antigen which can raise an immune response, while all the antigens may not raise an immune response. It depends upon the size of the antigen, the different kind of the molecular structure of the antigen, which we'll discuss in the later classes. All right. So coming to the recognition of antigen, so when we see that the antigens are very critical, we could see that there are microbial pathogens such as bacteria, viruses, there are other molecules as well, and there are very unique structures which are present on the surface of a pathogen. For example, here in case of bacteria you have a flagella. There is a flagella. This helps in bacterial movement. In case of viruses, we have the surface proteins which are expressed very uniquely on the viral surface. These are also molecular determinants which are present on the surface of the cell. So these are collectively called as pathogen-associated molecular pattern, which we have already discussed in the earlier classes to some extent. Now these are structural features. So immune response can recognize this pathogen-associated molecular pattern to produce an immunity. Now in case of the pathogen which are larger in size, for example, these are all microbial. We can always look these specific pathogen under the microscope, but there could be a pathogen which could be larger in size. For example, the parasite, the allergen, these are larger in size. So this may not be recognized by the body immune system as a specific entity. So most of the time their secretion and the structure which are secreted by this specific organism can be recognized. So we can call this as a functional recognition feature. So this can secrete certain kind of chemicals or enzyme that could act as an antigen, and the body immune response will be generated against those secretions or the functional feature which are recognized against those macroparasite. While in case of the smaller microbial organism, the structure is sufficient to raise an immune response. So there is a difference between microorganism and the macroorganism, for example the parasites and all, in terms of how the immune response has been produced, how the immune response has been generated. All right. There is another term which we need to understand called cognate antigen. Now the cognate antigen is something very specific to the types of receptor present on the cell. For example, here we see that these are the antigen A and these are the antigen B. Now the receptor which are produced by a particular cell is only giving an access to this antigen, not to B, and therefore the antigen A is considered to be a cognate antigen for this particular cell type, while opposite is true here, that antigen B is given access to this particular cell type because of the receptor which are very unique to this cell, while in case of the first one, the antigen A may not be recognized by this particular cell type merely because receptor is not cognate. So the recognition of antigen is largely depend upon the type of receptor expressed on the cellular surface. We have looked into this term called CD, cluster of differentiation, in the last class. So these CDs are very specifically giving access to the antigen into the cellular system in order to raise an immune response or do a signaling cascade to activate the cell population. So this biology is also very unique in case of antigens. We'll try to understand another example of antigenic variation. So here we are trying to understand a bacteria called Streptococcus pneumoniae. Streptococcus is a bacteria which is kind of a chain-shaped bacteria. So these are cocci, these are small spherical bacterial population, and these are in the form of a chain. When it comes in the form of a cluster, we called it staphylo, right? So this staphylo is either the bacterial population which are in the cluster, but here is in the form of a chain, and therefore these are called streptococcus. So in the streptococcus, the recognition unit, the pathogen associated molecular pattern which are present is actually the outer surface capsule. So these are the protein which are expressed on the surface of bacteria. These are polysaccharide which are present on the outer surface, and these are called capsule. So the capsular polysaccharide could be of different variety across this different species of Streptococcus pneumoniae. So here is example that there are many types of Streptococcus pneumoniae. For example, you see the structure is triangular. Here it is kind of a rectangle or square, spherical, again rectangle, and a different form, again different form. So there could be a multiple different when we are showing here in the color, we can indicate that the capsular antigens are different in nature. Although the shape is same, but these are all different kinds of capsular antigen which are secreted by or produced by Streptococcus pneumoniae. Now, for example, once the individual is infected with, for example, this first one, the body immune system will recognize this as a foreign and will raise an immunity against this. But in case when there is a second type of infection, when this particular kind of pathogen, the third number is coming into picture, then the body immunity may not recognize this as a foreign, or body system will recognize. So when there is an infection of a different variety of Streptococcus pneumoniae, in case this is the third one, then the earlier immune response may not able to neutralize this pathogen, and this is only possible because the structure of the capsular polysaccharide is changed. The earlier immunity was raised against these pathogen associated molecular pattern, but now the body immunity has to raise an immune response against a separate capsular polysaccharide. And therefore, once this new pathogen is identified as a foreign, the body will raise an immune response. That will take some time. But by doing this change in the capsular polysaccharide structure, this bacteria can dodge the immune response, evade the immune response. And therefore, the topic is immune evasion by antigenic variation. So this could be a very regular phenomena when a pathogen can change its structural pattern in order to escape the immune response of the host. Coming to the danger recognition model, and we have in lecture 3 we have discussed about the danger associated or damage associated molecular pattern. So these are specific proteins or molecules which are secreted by a cell which is undergoing some sort of damage. For example, we have discussed that a neuron which is damaged by certain toxin or some kind of a chemical reaction, or because of injury or because of infection by any pathogen, could generate this high mobility group box protein HMGB, and these are specifically a damaged associated molecular pattern. So this is called DAMP. So damage associated molecular pattern can bind to certain sensors which are present on the cellular system, and here, for example, this is toll-like receptor 4, and this binding of the toll-like receptor 4 with this damage associated molecular pattern can raise an immunity. So the inflammation will be generated, and this inflammation in turn will damage the neurons. So essentially, there is a cascade of danger recognition model that also exists in the host cell and host body. Coming to this same continuation, there is another phenomena that's happening in case of host cell, and that's called inflammasome related phenomena. So once anything which is called damage associated molecular patterns, once they are coming inside the cell, these cells can do a whole bunch of molecular signaling cascade internally, and there could be a damage to the mitochondria. So the mitochondria will release the reactive oxygen species. There could be a release of the chloride intracellular channels into the cytoplasm, and this can lead to the production of this NLR family pyrene domain containing 3 proteins, which are in short called NLRP3 inflammasome. So this is actually an inflammasome phenomena that is related to a body inflammation condition. So this inflammasome is produced in response to this damage associated molecular pattern. So there could be a possibility that a mitochondria release a lot of other molecules, including the potassium, and this can disturb the homeostatic biology or homeostatic aspect of a cell. Apart from that, these induction of NLRP3 can activate these caspase proteins. Caspase is actually a cysteine aspartate specific proteases. These are very important proteases that can cleave the protein into multiple different forms to do function. So there are caspases which are numbered 1, 2, 3, 4, 5, 6, 7 like that, and each caspase is having a very distinct function. There are caspases 3 and 7 which are linked to a cellular apoptosis, and here we are trying to understand caspase 1, which is actually a pro-inflammatory cytokines. So these are produced just before inflammation. So once the caspase 1 is generated, what it does, it activates the IL-1 beta, the pro form of IL-1 beta or pro form of IL-18 into IL-1 beta and IL-18. So these are pro-formed protein, and these are activated upon the expression of caspase 1, because caspase 1 may cleave the domain of this protein to make it active, and therefore the IL-1 beta and IL-18, which is an interleukin. So interleukins are actually the proteins which are secreted in between two leukocytes. Interleukin is in between two white blood cells. And apart from this, there is another cytoplasmic protein called gasdermin D. That's a very important protein that also gets activated with caspase 1. So upon the treatment of caspase 1, the gasdermin can convert into either its C-terminal form or the N-terminal form. So we can see that these two proteins are in bound form. Since it is a protease, it is bound to cleave the protein into two fragments. So one is C-terminal and the N-terminal. Now this N-terminal protein will go to the cellular membrane to simply make a hole, and through this hole the IL-1 beta and other molecules are passed, which can lead to the destruction of the cell type. So these are actually host response to fight against this damage associated molecular pattern. Therefore, we could see that there is a whole bunch of cascade to just stop the harmful effect of this damage associated molecular pattern. So damage associated molecular pattern could generate reactive oxygen species or the chloride intercellular channels from mitochondria, leads to the production of this NLRP family pyridine domain containing 3 proteins called NLRP3 in the cytoplasm. This can activate the caspase 1, and this caspase 1 can either cleave the pro-IL-1 beta or pro-IL-18 into active form of IL-1 beta and IL-18, and these are actually a pro-inflammatory cytokine. So they are produced before inflammation, and therefore they are called pro-inflammatory, before inflammation. And also the gasdermin D, which is a cytoplasmic protein responsible for making a hole in the cell membrane, can be cleaved by this caspase 1 into C-terminal and N-terminal domains. The C-terminal is doing some other biological effect by binding to some other cellular proteins, while N-terminal domain of gasdermin D can bind to the cell membrane and cause its polymerization so as to make a hole in the cell membrane. And through this hole, the IL-1 beta and other thing can pass through, so making this cell permeable to other molecule as well. And therefore, the cell is destined to be destructed by this phenomena. So this is again a very important damage related response in the human body. Thank you.